Hello, in this video we will discuss about the gustatory pathway. So what is this? This is the taste reception. During the sensory neuron will transmit the signal toward the brain. Affector nerves to the effector nerve will feel and not effector but it will directly move into the brain. So this is the person which that contain nucleus of solitary tract are present in the medulla oblongata. So this is the larynx, which also contain taste receptor, but tongue is major contain a thousand of taste receptor cell types. So in this way, here is the tongue, which that contains cell, and this is the brain. So in this way, here is the ventral posterior medial nucleus of thalamus, which that will detect the taste, either sour or bitter or sugary. So this is, let's begin to understand the cell generally of the tongue, which that contain a taste bud, and this taste bud contain a collection of cells which that will detect the different type of taste during the food intake. So this is the, for example, sugar will bind extracellular domain to the intracellular trimeric protein G protein, which that is contain a GDP inactivation to activate the alpha subunit will release from the beta and gamma, and the G protein which that contain GTP will activate the phospholipase C to convert the PIP2 into a IP3, and this is the second messenger inositide triphosphate from the from the phosphoinositide diphosphate, and when convert IP3 gated calcium channel will trigger the endoplasmic reticulum to efflux of the calcium from inside of the endoplasmic reticulum to the outside into the plasma into the cell cytoplasm. So it will trigger the calcium channel will open to influx of the calcium will lead to depolarization of the cell will lead to give the signal from the sensory neuron toward the brain. So I hope you make sense this is the seven nerves which that is, and the nine nerve and the 10 nerve, that is the cranial nerve, the vagus nerve, and we will discuss also with detail. So this is the ventral posterior medial nucleus of thalamus, which that will detect the taste, and this is the third order neuron. It is the interneuron to the third order neuron. So let's begin to understand. This is the front cross section of the brain, which contain a thalamus. Thalamus contain a gustatory cortex in insula. Basically transmit the signal to the gustatory cortex in insula, and this is the pons, and the pons, as well as here is the solitary nucleus in solitary nucleus in medulla oblongata, not medial but solitary nucleus in medulla oblongata. In this way, this is a tongue which that contain thousand of type of receptors, but the frequency of the sugar, the frequency of the bitter and sour, which that will detect differently. So that is why we will feel the frequency of sweetness and the bitterness. So this is the basically is the behind the tongue, which that contain which that is the vagus nerves, cranial nerve 10, which that transmit the signal sensation of the sweetness or something. And this is the glossopharyngeal nerve. This glossopharyngeal nerve is the cranial nerve 9, while the gustatory signal will transmit to the brain through a facial nerve, also the cranial nerve 7. So this all three type of nerve is the cranial nerves, which that transmit the signal to the solitary nucleus in medulla oblongata. After this signal, the detection, it will the second order neuron will transmit the signal to the thalamus, and thalamus contain a gustatory cortex in contain a nucleus. This nucleus is the center relay for the detection, and this third order neuron will transmit the signal toward the gustatory cortex in insula, and in this way I hope you get this concept. So anyhow, let's begin to understand. This is the tongue which that's contain a taste receptor cell. This receptor cell in the form of taste bud. This taste bud is open in slightly, and this slightly open the intact of the sugar or other bitter molecule or other acidic molecule will bind with the receptor, the receiving through a receptor. After the binding, the depolarization will be occurring to the cell. So how is depolarization? This is the sensory receptor cell, and this is the taste bud, and this is the taste pore. So in this way, this is after the detection, the effector sensory neuron toward the brain. That toward the brain is the effector nerve, which that is transmit the signal from the thalamus to the gustatory cortex in insula. So anyhow, let's begin to understand the taste bud. This taste bud contains different cells. So let's begin to understand the cellular mechanism, the signaling pathway with a different type of nutrients. For example, here is I am drawing collectively for understanding better with a different manner with a different type of nutrient. For example, amino acid, that is the protein. Amino acid basically the L-glutamate and glycine will interact with the umami receptor T1 or receptor 1 and T1 taste receptor 1 3. So this activate again the G protein to phospholipase C will activate the IP3 pathway, and in this way the IP3 will bind with the IP3 sensitive calcium gated channel will be bind with the open with the IP3 receptor, and after this binding to the in the endoplasmic reticulum will efflux of the calcium. When inside the endoplasmic reticulum calcium is too much high, but outside in the cytoplasm the calcium will be low. So in this manner the calcium efflux will lead to increase the calcium concentration into the cytoplasm, and the increasing of the calcium into the cytoplasm will trigger the sodium and potassium entry through a taste receptor PM5, which that will lead to trigger and depolarization due to the sodium and potassium will influx. Anyhow, the sodium will influx. So on the other hand, the potassium will outflux also. And in this way, let's here you can see this is the sugar, sucrose, and fructose. That is the sweet and artificial sweet, a sweetener, saccharin, which that will interact with the T1 taste 1 receptor 2, and on the other hand the K1 or 3 sweet receptor. When bind these things, and this will trigger the G protein collectively, and this receptor closely together, and that is why it is the basically the dimeric receptor, and this phospholipase C will trigger the IP3, and IP3 will again the same mechanism for the sodium and potassium entry through taste receptor PM5, and in this way the depolarization will feel through a nerve impulse will be occur toward the brain, and after this we can say that we are consuming the sugar and we can sense that. But anyhow, let's begin to understand the bitter receptor T2 receptor. So this is the quinine and denatonium salicin, which that is the bitter, and we can understand this is the bitter chemical, and the bitter receptor T2 receptor, which that bind with the G protein. Intracellular trimeric protein with G protein will be separated through a alpha G couple protein receptor, which that alpha will activate to phospholipase C again the IP3 pathway, and in this way the again the calcium efflux through same mechanism through IP3 receptor 3. And on the other hand, the calcium increase will sodium potassium entry through T receptor taste receptor PM5 will lead to depolarization. Will feel the bitter. While on the other hand, here is the acidic nature. Citric acid, for example, intact with the receptor here is containing the N terminal and carbon terminal. So the N terminal is the extracellular and carbon terminal is the intracellular. And so receptor PKD1L3, PKD2L1, which that will intact these all things. The citric acid acidic will lead to proton and potassium will be can be possibly influx will lead to depolarization. And on the other hand, here is the cation. Sodium, potassium, and calcium will also interact with the salt receptor ENaC or VR1 variant, which that will lead to depolarization. And PKD1L3, PKD2L1, which that will lead to depolarization, will feel the gustation, the taste